होईल ना. थांब थांब, तू याला पकड, मी इकडे येतो. थांब.
Good afternoon, everyone. Our esteemed faculty, Dr. Bhushan Gode sir, Principal Dr. Palikar sir, Dr. A.R. Ode sir, Vice Principal Dr. Nete sir, and all those who are joined through this online platform, I welcome you all on behalf of MGV Pharmacy College. This session that is webinar presented by our esteemed alumnus on fascinating topic of next-gen sequencing in pharmaceutical drug discovery. This session will explore how cutting-edge sequencing technologies are revolutionizing the development of new therapeutics, offering faster, more precise solutions. I would like to give the brief introduction of our today's guest speaker, Dr. Bhushan Gode sir leads the innovation development for pharmaceutical R&D. He received PhD in system biology and mathematical modeling with an interdisciplinary background of computing biology and mathematical biology from Oxford in 2006. He also had an interdisciplinary MTech in bioprocess technology and bachelor's in pharmacy in 1998. So it is a proud that he is our alumnus from batch 1998 and had been working across industry, university and government research institute with 15 years of experience in computational biology. In 2014, he joined UCB in the IT Early Solutions R&D team and was leading the data integration, image and omics-based data management and analytics-based drug discovery projects. His research interest involves biological data mining and visualization, mathematical algorithm design, high-performance scientific computing and simulation modeling of biological systems. In addition, he is always looking for disruptive technologies such as next-generation sequencing and post-NGS target validation. Gene signature-based mapping for drug repurposing and for target identification, artificial intelligence and machine learning for RWE, digital sensor data and biological imaging. With this brief introduction, I request Dr. Bhushan Gode sir to guide our students. Let's dive into this engaging discussion. Welcome sir, I will heartily welcome you from MGV, from the family of MGV Pharmacy College.
a couple of meters long, like meters long, and I have a photo next. Less than around 50 to 60 machines, and for the huge lab full of all these machines running 24/7 like a factory, like a factory. And they were generating these genome sequences for various organisms, from human, mouse, rat, cats. You can imagine. You can imagine. And that particular machine is what enabled the first human drug. But at what cost? It used to cost around almost, the estimated cost in that time was 300 to 400 to $1 million for sequencing one genome. The reason behind that was it involves a very complicated DNA fragmentation. So you know that genome in your body, in each of your body, each of your cell, a human cell, doesn't matter which tissue it's a certain blood cell. You have a nucleus, and in the nucleus you have this thread-like structures called DNA, and that DNA needs to be fragmented because you have DNA arranged into chromosomes, and these chromosomes need to be fragmented, and the DNA fragmentation, when you do it, they need to be clonally amplified, so there's a clonal amplification involved in. So you insert them into a vector, which is some kind of a microbe or something, and using the cutting with the enzymes, and you know it by biochemistry. You learn these enzymes which cuts this DNA and then joins it, and then you insert them back into the cell. You grow the cells, and then you amplify those, so you can get the DNA, particular DNA fragment amplified, and then you do a whole capillary sequencing. So this is electrophoresis capillary sequencing, which took a long time to basically perform these analyses, and exactly that particular human sequencing step was automated by using the Sanger machine. This was the Sanger machine, which used to do this kind of Polanyi array. So this is like a Polanyi of a small amplified genomes, and then of your fragment, and then the fragment will be then photographed around, and then you will be manually matching which of this fragment match to this particular dot in a manual way. And that example is the Sanger sequencing, and that was the first machine, automated machine, which allowed them to do sequencing. But it used to take still a long time. I mean, it used to take months to sequence one genome, because you have to do a lot of fragmentation. The fragments cannot be more than, say, 60 to 100 bases. You know that there are 2 billion bases in our genome, so you can imagine how many such fragments need to be there. And then there will be regions which basically on a chromosome, it will be on a telomerase end or centromerase end, and they have a lot of repeats. They call that the same kind of a sequence gets repeated many times, and it's called alu repeat. Nothing to do with our alu vada, but they call it alu ALU repeat, because they get repeated many times, and these repeats are very difficult to sequence because you don't know how many times that repeat is happening in that particular region. So such kind of challenges, whether I used to call that in my standard time case, these we used to label it as dark gene, because you cannot really see what is going on into this particular telomerase and centromere region, the center of the chromosome. And that was a challenge back in the first decade of around 2000. And so even though Sanger sequencing is a revolutionary technology, it really, let's say, it's a stepping stone into a sequencing of human genome. And it what revolutionized it to the industrial scale what we have today. So that is the reason we should learn about Sanger sequencing. By the way, anybody knows why the name Sanger is given? Sanger, the Sanger name is in the honor of a great scientist, Professor Dr. Sanger, who actually helped in identifying the chemistry for this particular capillary electrophoresis? How can we bind these particular template primers to identify the genome sequences or the fragment sequences? And he was alive when I was working there in 2008. He used to visit a lot, even though he was quite old and retired, he used to visit. And that's in the honor of his name they gave the Sanger Institute Sanger Genome. And this is exactly the photograph I took back in around 2009 when I was working there, 2008. This is the Sanger genome sequencing. And then what happened was that revolution takes place in technology. And so as I said, the cost of whole genome sequencing for one human was around 1 billion to 10 billion, anywhere in between. You don't know how many times you have to repeat it, how many times you have to correct the errors, etc. And then what happened was there was a company who used to manage these machines back in 2000 in Sanger's Genomes in Sanger Institute, and that company was a spin out of Sanger's research, basically research frame, and that company was called Solexa. And then Solexa was in a Cambridge, a small startup, and that Solexa came up with the idea of miniaturizing what Sanger is doing, using a modified chemistry by changing the way the reagents work, so the capillary electrophoresis work. Instead of being a capillary, they made it into a horizontal, let's say, flow cells, and by using a flow cell, they could make a machine which was as small as, let's say, a box, you know, which you can put it on your tabletop. Let's say you have in your lab, you must have seen some kind of, GCMS or mass spectra or something like that, a small ball. So of that size, they could able to make it, and then they changed the name back in 2000, let's say 8, from Solexa. They sold it to a company called Illumina, and that sequencing became popular, called Illumina sequencing. Now, Illumina sequencing was very high throughput. It could do a genome sequence of a human genome, let's say 24 hours. We just prepare the samples, prepare the lab and laboratory DNA extractions, and then put a reagents and put it into the cell, and then there was a flow cell which will basically complete this kind of sequencing, and it will cost around, let's say, 5 to 10,000 per human genome, dollar, depending on what machines you are using and how many mass manufacturing you are using. So that was the first generation of NGS technology, and that basically brought down the price from let's say $1 billion to $10,000, and that was quite a big. I mean, most of the pharma started buying that time these kind of machines in their laboratories. Most of the biotech started doing these Illumina machines purchase, and then they started sequencing. Then the second and third generation started coming. So this was the first generation, this was the second generation, and then the third generation started coming. Well, then they started miniaturizing, and there were companies like Iron Torrent and Oxford Nanopore. So this Oxford Nanopore was a really technology which we can actually sequence the genome onto your USB drive. So this is a USB drive, you can just put it into a computer, and you don't need a machine which is of the size of half the room or something like that. You can just do it into your laptop. You just put the sample which you prepare into the lab, into this Nanopore's mini-on machine, and then within, let's say, a couple of hours, you will have a sequence in your laptop directly via these USB connections. High-speed connections, you could actually get the sequence and alignment onto your laptop. And it reduced down the cost to, let's say, around $1,000 or less for one genome sequencing. Now, you know how much $1,000 is today, around 70,000 rupees. In rupees, you can actually get your genome sequence full, including your intron exons and whatever, the DNA, mitochondrial DNA, etc. that can be sequenced now. So this is the revolution that we are talking about. When we say there is a revolution happening in certain areas, not only the speed is increased, not only the size of the machine has reduced down, what you can carry with you, the amount of time to process the genome sequences, and the amount of errors introduced involved in sequencing. So there are many errors involved in this particular sequencing, and so that's why you have to do hundreds hundreds of thousands of runs again for small fragment to see that how much is the consent for correcting those errors versus there is no error reported into this particular sequence because it is using the chemistry which will be keeping long sequence generations and record accurate. And also these machines are now possible to like carry with you. So we have a lab in Nashik and we do sequence. So if anybody want to see this machine, I can give you the address and you can see these machines working there in Nashik itself. These machines are the Oxford number machines that will taken to the space station, International Space Station, where they can actually sequence the genomes of whatever planet they go, whatever experiment they can do. So in space studies, these are the machines they actually use it as well. So that can be the way to improve the genomic sequencing effort there into the space sections as well. So that's the equation. So what happens in this particular technology? Will basically this graph is showing that as the time progresses, the cost of the sequencing is coming down. Will it go beyond this? Is a big question. I mean, this slide I made in around 2016, 2017. So I don't have predictions about that. But what we see now, after 10 years of this slide is made in 2025, if we see here, the cost hasn't gone down too much beyond, let's say, 1/5 of it, so $5,000 to now $1,000 or something like that. But we need to see if it goes more down than this particular equation. There is also, so we were in the genomic era when we wanted to sequence the genome. We are now in a post-genomic era where we have hundreds of thousands of species have been sequenced by various labs, various institutes around the world. And now we have the data, we have the databases around to look for those genes and their sequences, the RNA, the DNA and the RNA equivalent to that. And so what can we do? What can we see about these databases growing? And so these are the projections that if we apply the Moore's law, it says that every 18 months the cost of the electron history devices reduced by half so kind of rather than doubling time, it is half. So that we can see that the growth by doubling every 18 months, we can actually reach by 2025 almost the number of human genomes sequenced on the Earth, so on the population, would reach to let's say, 1 million genomes would have been sequenced. And I can tell you that this is true now, that by 2025, we have more than 1 million genomes sequenced only in a small country like UK. I don't know how many sequences are there in other countries like America, Europe, and developing countries like China, India, and others, but there are more than. So instead of doubling time by 18, I think we could see that doubling every 12 months is what estimated, that we actually have more than, let's say, a few million genome sequences are now available on the Earth for human genomes. And so this is the graph which projected that. Let's now dive into why these different platforms and how they work. So there are various platforms, ingest platforms, and maybe I'll come back to this slide after I introduce to them first. So let's see what are these machines and what these machines are. And then when we're talking about second and third generation sequencing. We are not talking about the Sanger genome sequencing because first generation is almost dead now. Even Sanger doesn't use the Sanger genome sequencing anymore because of the cost of running and cost of maintaining. This all is replaced by now. They live now high-sec, MySec machines, which are the next generation machines here, and you can see it in the photograph. These are the machines. So let's start with the first company. After the revolution, at the same time, there was a company called 454, and it was purchased by a pharma company called Roche. So it was famous as Roche 454 sequencing. And this Roche 454 sequencing was revolutionized in a way, but it was a pyrosequencing. We used to have some kind of a pyrosequencing technology. If you need it for the mechanism, you should look for YouTube videos of those corresponding for the mechanism of them. But it was, again, genome sequencing. It used to do very small fragments. I mean, when I say small fragments, it's like around 300 to 400 bases of genome will be sequenced and not more than that. Then came along with that called iron torrent. And iron torrent was based on some electric signal based sequencing. So whenever the base kind of get amplified, there is a current release, and in that current they call it ion current. Because each time the base is consumed, there is a particular ion will be released, and that ion will be captured to measure which base was selected, whether A or G or G or C, and that based on ion torrent machine, and it was quite famous sequencing for fixing those errors which will be not fixed, be able to fix by other sequences like Illumina and 4x4. So ion torrent was one of the good technology that they were using. And then again we saw the Illumina MySafe, which was using the flow cell technology, where they actually let the cell flow along with the area labeled bases, where you have ATGC flowing through the cell, and then we get binded to a DNA to get amplified DNA. And then every time a particular DNA base binds, it illuminates the signal, and the signal will be captured, and then that will be measured by taking hundreds or thousands of photographic analysis will be done on top of it. And so then there was an ABF solid, so there was a machine called solid, and there's a long form I think involved in there, but this was purchased by ABM. And that ABF solid was also a good industrial standard machine that used to sequence. And then the Illumina had MySafe, which is for small labs, pathology labs, and small hospitals and marine labs. But for larger scale, for bigger scale, they created HiSafe and other TrueSafe and other sequencing, larger machines 24/7, and so they have HiSafe and MySafe and TrueSafe machines. Then there was a Pacific bio sequencing. It was like for small genomes like microbes and viruses, identifying some kind of a target, etc. That was used. And finally came nanopore sequencing, which is the mini or the USB disk-like sequencing, which was developed by an Oxford-based company called Nanopore. And as the name suggests, there is a nanopore involved into a membrane, and that membrane will allow the genomic sequence to, so double-stranded DNA will be guided through that particular pore, and then one strand will be eliminated, or one strand will be denatured, and only one strand, either lagging strand or lagging strand, will be able to pass through that pore. One base at a time. And as the base passes to the pore, that will allow the change in basically electric current variation, difference between the potential between the two sides of the membrane will be measured. And that signal will be used to identify which base has just passed through the pore via the enzymatic pore or nanopore. And that is exactly how the nanopore works. And there is a good mechanism involved. If you are again interested, you should look into all these YouTube videos on the YouTube. You don't need to look for the links. Remember, you just remember the names and you just search for them to YouTube, and you should be able to get their mechanisms, etc. Now, the applications of these were quite important. For example, when I was in GlaxoSmithKline, GSK pharma, working for GSK, we used to sequence the aim of the GSK was to manufacture amoxicillin as much as possible using the fermentation-based method. So we were using all these Streptomyces, the strains, and the strains need to be improved. The strains will need to be always kind of improved for production. So how much? So let's say 1 liter of fermentation will give you 10 milligram of amoxicillin or penicillin or kind of other antimicrobial production. Can we do that for 1 liter to, let's say, 1 gram, improve the yield? And so for improving the yield, how can we improve the yield? And that time we used to do this kind of genome, targeted genome sequencing based mutagenesis. So we know which are the pathways responsible for producing the particular, let's say, antimicrobial antibiotic. And so can we enhance the production by, let's say, modulating those genomes or increasing their expressions into the newer cell lines, and then improve the cell etc. And so that particular experiment could be done earlier, while random mutagenesis was the norm before 1990s or 2000s. Now what we do is we know the genome sequence of the strain from the beginning to the current state, and then can we use those studies to improve it. And these are the methodologies we use. We could do actually the RNA sequence into RNA, and RNA is what is responsible for causing the protein expression. So proportion to the RNA expression, you will have your, let's say, condition worsen or condition improved kind of a scenario. And that's what we used to do by using this sequencing. We used to check RNA sequencing, how much is a particular gene expressed in a disease condition versus a healthy condition. Let's say in a cancer state, which are the triggers for cancer compared to healthy particular cells. So we can see the differences between the two patients. Patient and the healthy person, to see which are the responsible target site or gene site. And then similarly, there is chipset analysis and a whole set of various DNA seq and other kind of sequencing that we could perform into that. There's also the epigenetic studies, where we know that certain genes have separate mechanisms, like lac-operon is what you study in your life chemistry, for example, which has controlling mechanism. When the sugar level in the blood goes to high level, the operon kicks in, and then the expression of the gene starts, and then RNA forms, and then the sugar kind of regulation mechanism starts. So exactly like that, lac-operon is the simplest mechanism. There are much more complex epigenetic mechanisms in our body that is involved in immune inflammation and other kind of day-to-day functioning of our body. And that epigenetic markers will be isolated out to be studied using this kind of genome sequencing. This is just technology's landscape and the comparison side by side, what it is and how much basically strength and weaknesses of these. So for example, nanopore gives you up to 100 kb of a DNA sequence or RNA sequence into one single, because the core, once you started getting the ratchet mechanism, like a zipping mechanism through the strand of your DNA, it could not stop until 100,000 bases passed in. So 100,000 bases is a long sequence compared to, you can see that Illumina, even with the improved chemistry, was only up to 600 bases. 454 only 700 bases. Well, I told you, the whole of the genome is 2 billion bases. 2 billion is quite a big number, by the way. And I don't know how many of you know how many zeros are. But 2 billion versus these kind of 500 or 600 bases, it will take you a long time to map them back to get the whole chromosome map of your DNA and mapping it. So you can see that no other technology compared to backbio and nanopore will give you that kind of a long thread of DNA in kind of an identified. And so that will be the that's the reason I call them third generation sequencing, because they are improved sequencing. That doesn't mean that they are very accurate, but there is some kind of error type involved in their measurement. Because we know that they have an indel insertion. So indel is insertion deletion error. So when there are multiple poly A's or poly bases basically in the sequence, so there is a situation when your DNA has 5 or 10 adenosine. What happens in this particular machine is it kind of doesn't give you exact 10 number. It could give you 11, it could give you 10, 9 or 8, because it misses the current amplification. So there are these kind of errors involved into each of them, and there are biases that are according to certain machines that we are looking at. But again, given that how much it costs for run as well, there used to be various numbers. The reach per run can be variation, and these are high throughput runs. So you can see that there are 10 gigabyte of bases can be generated by one run, whereas the run is in Illumina is now nanopore is improved to similar kind of capacity, still slow through. Also, what I didn't tell you about this nanopore is this machine, nanopore can be recycled, but inside the machine the membrane, there is a cassette you need to put it, like audio cassette, how many, I don't know, how many of you lived into the era of audio cassettes, like you used to get audio cassette back back in few, in early 90s, or 2000, like exactly like that, the cassette every time you do the experiment, you need to put a new cassette in here to get this sequencing done. While Illumina and all, there won't be any cassette, they will be using the same machine at a time. So that refill needs to be done and it needs to be purchased, so that's the additional cost for them. Yeah, there are some advantages and disadvantages we discussed, and the basis, there are per billion, so 1 million bases will be $3 cost for these buses compared to Illumina will be lower, 0.5 for 1 million bases, $1.05 or something. So there is advantage and disadvantage in terms of price versus sequencing technologies there. So why are we studying this? Why are we doing this? Why I am telling you all this thing? What's the use to pharma drug discovery? So pharma drug discovery, you can see the schedule on the top if we classify into key areas. Let's say the first area is target identification for R&D. Then second is target validation, then lead identification, candidate optimization. These are the key stages as we progress the drug from one phase to another phase. And then preclinical and clinical studies, and that's where, and then even there is a post-market, kind of a post-market, which we don't show it here, but it is always involved in, and that's kind of thing. So in all of these studies, the NGS sequencing plays a bigger role, from identifying, validating this target, whether what we think is the gene responsible for a disease, is it right gene? Is it really the correct gene that we are working on? Is it something else? Is it the right target? Is it the right validation that we need to do? Then we check with it healthy and versus the patient population to see is that the gene which is get affected because of its, let's say, genetic error or whatever change in mutation or something, or what kind of, let's say there are around 10,000 rare diseases, and out of this rare disease, around 60,000 rare diseases, 6,000 rare diseases are genetic. And out of genetic means what something happened at the time of birth or before the birth, and that is permanent problem in this particular people in hemophilia or some kind of blood disease like that happens like that. And so these diseases need to be checked whether which of the genes responsible for it. Out of 6,000, by the way, almost 3,000 are monogenic disease, means only one gene, one error somewhere in that particular gene has caused the disease. And the rest of us are multi-gene. There could be 2 or more genes responsible for causing that particular disease. So these kind of validation need to be done. This kind of disease etiology need to be identified. And so that involves studying the population and identifying the real cause of the disease. And so that is the pre-discovery phase. Then we need to identify, once we identify the site on the genome, we can see how can we can we edit. And so I don't know how many of you have heard this for CRISPR-Cas9. So CRISPR, I haven't touched this particular topic of CRISPR-Cas9 because it's totally, it needs another hour or something to explain. But these are the molecular scissors that you can actually cut your DNA, which is bad or has the mutation happened. Either can be a single nuclear polymorphism, or could be update insertion or deletion in your genome, and that can be fixed by using CRISPR-Cas9 and cutting scissors and fixing them. And so that's the reason we need to. But to use this CRISPR-Cas9 studies, we really need to have an accurate map of your genome, of the genome of the patient you want to treat. And that is really must, because otherwise if you don't have the real map of the patient, you are going to do some kind of rather than helping his to treat the disease, you can actually cause the damage into his DNA, and the DNA damage will be catastrophic, and it will stop getting cured. It could be lethal for the patients. And so these are the reasons that we need to really study the particular genome sequence of the patients. There are many other experiments that we need to do sometimes to identify whether the drug is working or not. So is it actually effective? Is it controlling the conditions? Is it a molecule that we are considering as a drug? Is it really fixing the problem? And so that we need to go and check onto the population and see what is happening. post-treatment, or is it really optimizing, is it really fixing the problem that we have. And so there we need to do the study by using early expression, epigenetic marker studies, etc., to see that the drug has acted upon and what we are getting is the correct effect or not. So these are the challenges, these are the actually use of the next generation sequencing into the early drug discovery. The same thing is in the clinical phase. We have various clinical phase 1, clinical phase 2, clinical phase 3, marketing, authorization, etc. And all this needs to be sometimes validated using the FDA or regulatory bodies saying what we are being giving is helping making a sense to the patients and population that we benefit. This is exactly the same diagram in this is from my own paper in 2021 on the application of sequencing, but what we see here is exactly the diagram picture form of this chevron that we have been showing you. So from gene to target screening of compounds, so this is the compound screening library, screening of compound candidate, selection, control selection, first time into human, POC, and then first time POC, phase 1 to 3, study starts actually, and then phase 4 and marketing and post-marketing. And so each of these blocks here is application of the next generation sequencing. That blue blocks are basically telling you what technology we will use in that particular period, particular phase to identify the effects, etc., that we could study. For example, in the lab models, mouse mutant can be helpful to study whether the particular gene block or gene silencing is working or not, or particular gene reactivation is working or not. So you can create a mouse with the mutant. You chop the particular gene from the mouse and then use the drug to treat him so that you can see the effect in it, etc. So lab mouse could be edited, edited for various tests, etc. Provided the biology of that particular gene active pharmacology is same between human and mouse, which is not the case in most of the cases. But lab mouse is very easy to handle, so that's for some of the tissue that we generally do the engineering for. And the last thing, so back in 2011, this paper we wrote. After we wrote this paper, actually in 2012, there was a big paper published called ENCODE. And this ENCODE is nothing but the epigenetic marker I was talking about. So this is how your DNA actually is in the cell. So this is your cell in the nucleus, and then there is a DNA coiled into chromosomes. Of course, each chromosome is also coiled in, and then if you try to straighten them, what you see is a thread like this. And this thread is not really look like a thread. It still has some, let's say, stones, 4 stones put together. These are the stones that are together like a barrel or a dumbbell. Around the dumbbell there is 1.5 or 2.5 turns of your DNA going on each of these dumbbell barrels, and then the 4 barrels are put together to kind of look like some kind of coiling mechanism. And so this is a beautiful arrangement of packing the dense packing of the DNA into a cell. You don't have space. Basically, if you stretch your DNA after joining all the chromosomes, it will be around 3 to 4 meters long, 3 to 4 meters, and that's a big distance by the way. That's a kind of a small thread it will be. And that thread it will be packed into a small cell which is less than one, let's say few micron thick nucleus, and so that's beautiful pattern there that will be. But the idea about that particular packing is also to access those features, they call it non-coding elements on the DNA. So DNA as such or genome is arranged in parts, various kind of islands, and the islands are introns and exons. You have studied these in your biochemistry or microbiology, and then introns and exons. So what was thought about that is this. Basically, introns are just the gaps between the exons, which are actually makes your RNA. When they express, they make your RNA, and they're just the gaps to protect your DNA from damage, etc. Turns out that it is yes true, but it also causes alternative splicing. So, for example, a particular gene A could have 3 or 4 different splice variants. It will give you different proteins and it will work in different conditions. So, combinatorial possibilities are also there. But along with that, there are many other regulatory elements. Like I said, lack of it was one of the simplest ones we knew. back in 2000, earlier, let's say early 1960s, 1970s. But then there are many such markers were now found, which are nothing but it sits into this space called jump, 90% jump of the genome space. And they were thought about jump, but they are not jump. They actually regulate from being there to the expression of the gene. And so that is what we call the genome about the gene. And that genome identification was possible because of this NGS sequencing. And that is what is revolutionized the way how we kind of study this. And this particular field now is responsible for many discoveries into pharma industry, especially in the area of neuro and neurological sciences, even pharmacology, especially immunological diseases, and last but not least is oncology, basically, where we understand now why certain cancers have such a prevalence, etc., high prevalence in that. With that, I would like to stop here and we take a few questions. Unless there is more questions, let me know. Because I can see there is a 31 and I would like to give some time for students to ask the questions.
If anyone has questions, they can go or write in a chat box.
If anyone has any question, they can write down in chat box or they can unmute themselves and ask the question. All students, those who join the session.
I can read it for everyone. It's from Bhavesh. How has the field of sequencing evolved from first generation to third generation technology? It's a good question. As I said, this particular thing is led by this particular generation. A revolution was led by the competition, the demand for the sequencing. Sanger sequencing has a big demand, but big money involved, so it was hindering the discovery, biological discovery of genomes, etc. There was a huge pressure, demand, etc. What happened was really the evolution kicked in because people working in this area found out that this could be improved. This particular machine, which has, let's just say, 2 meter, 3 meter by 3 meter, kind of a big cubic box, the technology there could be improved, and that happened by 3 different ways, by the way. I mean, first thing is people challenge the way the things are being done. That's something I think Indian students don't. Okay, you do not challenge whatever you say. Okay, this is it. This is how it is. First thing is challenge the current status quo. Don't say, okay, this is how we do it for a long time. This is how it's meant to do. No, challenge. And so people started challenging the way. This is the right way to do it. That made them think that there is a better way to do it. The second thing that happened was the technology advanced, the understanding of, let's say, chemistry behind what is happening inside, the electrophoresis, etc. That chemistry, the new reagents, binding, etc. of the bases to the new chemical structures, the eliminated fluorescence, etc. binding to these dyes, etc. has really revolutionized the way people could do this. And so instrumentation analysis was another area where there was advances going on. You see, GCMS, mass spectra, and all these instruments have been also improving over time. So these companies also started putting into effort to say, can this be done better, can this be done robustly? And the third thing, I think, that really caused the technology started exploring other areas of physics and chemistry and biology. Basically, for example, nanopore. Nanotechnology was emerging in the first early decades of 2000. There was no technology before called nanotechnology. People started doing microscopic analysis of the compartment, saying how big is this particular nanopore there, hole is there, what can be done, can we use this for some applications, etc. And that really caused application diversification, exploring these particular technologies beyond the traditional chemistry. And so these caused from first generation to second generation to third generation. And I mean, this is just an example, by the way. The iron torrent wasn't using any kind of, they were using what a chip mechanism, like your Intel and Silicon chips are there on your supercomputer, on your computers. These particular chips has all this device, these particular fluids going on. So that kind of devices they were using microfluidic channels, which are passing all these things. And you can see that the image here on my screen is resembling to that photograph of your Intel chip like photo. The chips there was an area called lab on chip technologies that also revolutionized this particular sequencing. So people were thinking, why do all these things, experiment we do in animals, etc., into the lab? Why can't we use miniaturization? Just take the cell, one cell of a rabbit's liver or something, put it into a micro pipette into a glass slide, send a drug in, identify what is happening to that particular cell, rather than putting on the whole mouse. That's the advantage. That's the revolution. And that is what it's called. So there was a full field called lab on chip technologies, where they say you don't have to do all this big lab experiment, we just do it everything on the chip. Chip means a glass slide, and we can see in microscope what is happening. That kind of technology is really revolutionary. And I don't know whether these technologies will be fourth generation, there will be fourth generation possible. We don't know. You guys will do fourth generation. You need to study this with this particular passion that challenge the current status. Say Oxford Nanopore has problem. Yeah, there are some problems still there. The cost is still high. The dream was to get a genome sequence of each patient in $100. $100 is like something like 5,000 rupees. That is a dream. We haven't achieved that dream yet. And so that dream, until that dream is there, when 5,000 rupees, the genome sequence will be there, I'm sure the government of India can do each and every person's genome sequence in your other card. I don't know. Something like that. So I hope I answered your question, Bhavish. There is always limitation. Yes, any technology has limitations. Basically, as I said into a slide here, slide over there. Yeah. So you can see, as I said, every technology has its own challenges. No technology is foolproof. What we used to do in fact, and that's a good thing to tell. Because we were in drug discovery and we want accuracy, we want 99.99% accurate of our sequence. We couldn't get accurate sequence of, let's say, my streptomyces species, when I was sequencing streptomyces. I was sequencing human and this microbial streptomyces. Streptomyces genome is not that big, I think 5 MB to 10 MB. So that's not that big megabytes compared to 2 gigabyte or 2 gigabyte of our human genome. So it's a small genome. Still we have a lot of errors with Illumina sequencing because we only get of that 5 megabytes or 10 megabytes, we get only 600 bases and you have to map those back into this particular. So we do big sequencing. We need Illumina and 454 to eliminate the errors. 454 has errors, Illumina, but the errors are different because of different things. So when you merge together, you will nullify the error by each other and then you will get a better sequence. And that's how we used to do that this is back in 2010. But now in Nanopore, you still get errors. And so you have to do that targeted sequencing to remove that error, etc. So yes, there are challenges associated even today with these technologies. And again, the biggest challenge I just explained to Harish in the last kind of point. The biggest challenge is the cost. Can we reduce the cost down to the full human genome, something like $100 or like $50, 2,000 rupees, like 1,000 rupees. That's the challenge we need to. That's the biggest challenge.
Anyone has any question? Other students? Thank you very much, sir, for such a great talk. Now I request Dr. Katti Madam to propose vote of thanks for today's session.
Thank you, Bhosle Madam. I wholeheartedly thank Dr. Bhushan Gonde sir for his informative session on next generation sequencing in pharma drug discovery. Sir, your lecture has given a detailed information about the importance of genome sequence and the latest development in genome sequencing. This has motivated students to know about the latest development in genome sequencing and its application in pharma industry. I thank Principal Dr. Tambe sir for motivating us for keeping and arranging such sessions. Sir, you have spared your valuable time for our students. So thanks for your cooperation. You always arrange at least one session each year, which really motivate our students to take such projects for the research and also motivate for take such projects for PG and PhD and apply for the institutes for such research. I thank the participants who have joined this session by online mode, and the thanks to TYB Farm students who have helped us to have the live streaming of this session. Thank you very much, sir. With this, we conclude the session.